So next we're moving on to methane. Our first speaker is going to be Ronimus from AgResearch, and he's going to join us virtually. He currently leads the PGGRC NZACCC methanogen inhibitor program, and has been working his way through an incredible number of compounds and assays. Ron, are you ready for us?
Methane inhibitors for use in pastoral grazing systems. I would like to introduce the team. So I got myself and Dr. Vince Carbone as structural biologist, Dr. Lindley Schofield, who's an enzymologist, Dr. Peter Jansen, who's the microbiologist, and Dr. Stefan Mutzel, who's an animal trial specialist. At the University of Auckland for chemical expertise, we have Dr. David Rennison and Dame Distinguished Professor Margaret Bemble, plus their postgrads. At the Dairy NZ, we have Dr. Elena Mani. At the PGRC or /RGP, we have Mark Aspen. For the commercialization team, we have Dr. Ian Boddy, who's a chemist who handles commercialization affairs, and Dr. Jane Calvert, who's also a chemist and handles IP issues, and Catherine Kimshaw, who helps us with regulatory affairs. At Argenta for capsule development, our main contact is Dr. Desmond Morrow. For developing slow-release capsules for pasture-fed animals, we're basically aiming for 30% inhibition that could last around 200 days. The current technology would get us up to a maximum of around 320 days. Slow-release capsules require highly potent and stable inhibitors that can be formulated into the tablets of the capsule. Any compound suitable for capsule use could also be used as a feed supplement, sort of goes without saying, or other delivery system. The capsules allow us the tailoring of dosing levels, so for instance we can lower the concentration at the beginning to aid adaptation to the rumen. Now for capsules lasting 200 days in sheep, the payload is 7 grams, which equates to 34 milligrams per day. In cattle, the payload is 52 grams, equating to 260 milligrams per day. Our current data suggests that about 2 times as much may be needed using capsules to achieve the same 24-hour level of inhibition compared to when a compound is added to twice daily feeds. The main reason for this is that the capsules release 24/7 and methane emissions are lower at night. Now, an earlier lead at 30 mg per day in sheep trials, dosed in feed, caused 30% inhibition. That same compound, when dosed by a capsule at 39 mg per day, caused only 7% inhibition, but this was using an artificial ad lib pelletized lucerne diet at 2.5 times maintenance. I would now like to tell you about our methane inhibitor development pipeline. I'll spend a couple minutes on this. On the left, we have discovery, so we look at methanogen genomes to find enzyme targets. We then purify those enzymes and develop assays. We also have a methanogen pure culture screening assay, which we use. We have a rumen fluid in vitro assays. We have two of those. We have a micro method with 0.2 mL and a larger method with 50 mL, which I'll tell you more about later. We have an enzyme in silico screening technique, which is basically docking compounds by computer into the active sites of enzymes and scoring them. And then we also use enzyme substrate analogs to develop inhibitors. Once these gets hits from these types of screenings, we then want to go to optimize the compounds, and we do this through medicinal chemistry at the University of Auckland, or we can sometimes source analogs of the compounds. When those come in, we retest them in our various assays, enzyme assays, pure culture assays, or rumen in vitro assays. And then the best of these derivatives will then be ready for in vivo testing. The first protocol there is a mouse toxicity test at 300 mg per kg, which is basically about 50 times higher than anything we would use in a ruminant. This is a safety trial. We then are able to go into short-term chamber trials in sheep for 3 days. This tells us if the dose is right or not. If the dose is right, we can then go into a midterm sheep trial, 16 to 28 days, and there we want to see if the rumen adapts to the compound at all. Once we have that data, we're happy to go into cattle trials. Usually the first protocol there is chambers, which is more accurate, again for about 2 to 3 days. And that's also to make sure that the dose is right. If we're happy the dose is right, we can then go into a midterm chamber trial with cattle or use a green feed system. And if that works out okay, you're then in a position to do longer term trials around 90 days, something like that. And during this times, we're also looking at developing capsules for use in some of these trials as well. So not just using feed supplements. And then, of course, we try to blend these studies into regulatory approval studies. Right, so methanogens are archaea, and as such, they have rather unusual features, including unusual lipids and cell walls and some essential metabolic pathways. But here we're concentrating on methane formation. So we have carbon dioxide and hydrogen come together to form methane in an 8-enzyme reaction sequence. This requires 6 specialized cofactors, which are in the boxes on the left: methanofuran, methanobutyrate, F420, F430, coenzyme B, and coenzyme M. We have solved 13 structures from these processes, which is quite helpful and can be used for finding inhibitors. On this side, we look at the targeting of enzymes required to obtain fully active methane-forming enzyme, which we call MCR. This involves 3 methanogen cofactors, F430, coenzyme M, and coenzyme B. F430 is special in that it's got a highly reduced nickel ion that's absolutely critical for the reaction. The formation of fully active MCR also requires an additional 16 enzymes for the activation, which is basically involving the reduction of that nickel ion, the assembly of the subunits of MCR, and the post-translational modification of the active sites of MCR. We have solved 12 enzyme structures relating to these processes, which can also be used for development of inhibitors. This slide here summarizes our enzyme pure culture and rumen in vitro screening assays. On the left, we have a 96 well plate with our culture assay. In the middle, we have the 50 ml rumen in vitro assay with 32 bottles with automatic gas sampling, which can produce graphs similar to those on the right, with this showing methane on the left and the hours on the bottom axis. The top red curve is the control curve, and then those below that are in the presence of an inhibitor showing inhibition of methane emissions. Overall, we screened 140,000 compounds against various enzymes, 8 enzymes. Over 40,000 compounds have been screened against methanogen cultures. This equates to 15 million compound enzyme interactions. We screened over 15 million compounds using our computer-based assays, and we've screened approximately 13,000 compounds in duplicate in our rumen fluid assays. All these strategies have produced hits, about 1,000 overall, validating the approaches that we've used. I'd like to give you an overall summary of the discoveries of the pipeline and some current work. All those hits I mentioned ultimately were grouped into 12 classes of compounds, about half of these with over 100 analogs each. And we've tested 40 plus compounds in animals. Our current lead class has over 140 analogs, and I'll give you more details on that on the next slide. We have screened a 10,000 compound library, which some of those hits have shown good inhibition in the pure culture assays. These could lead to additional new classes. I'll just make the point that drug discovery requires an ongoing pipeline approach with a steady flow of compounds for testing and discovery of new hits, followed up with medicinal chemistry optimization. So on this slide, I just wanted to focus on this new class of inhibitor that I mentioned. The first compound in this class was found in one of our screening assays, and an early derivative has been tested in sheep and led to 11% inhibition. Our latest derivatives are up to 30 times more active in vitro than the first hit discovered. Some are active at low nanomolar levels in vitro pure culture assays. And we've recently tested 4 examples of these compounds in sheep, which worked better than we expected, which was good. The understanding now is that there is likely to still be room for further development within this class, using medicinal chemistry techniques to help us potentially achieve the target dose for capsule delivery. So we have other research ideas for finding and delivering inhibitors. These could include expanding work on backup classes from the pipeline. We could systematically target the 6 methanogen cofactors using substrate analogs. We could continue screening large compound libraries using our methanogen pure culture assays. we could expand the use of our in-house enzyme crystal structures for identifying new inhibitors using computer-based screening methods. And always we're looking for delivery mechanisms. This includes adding inhibitors as feed additives, use of capsules, of course, which is the main interest, water delivery, and perhaps even things like salt licks. And of course, we will ultimately want to scale these studies up to for use on farm. Right. I would like to thank some people here and organizations. We'd like to thank the Australian Synchrotron, the PGGRC, the Greenhouse Gas Center, the New Zealand government, and Marsden for funding. There's been over 100 people involved in this work in the last 10 years or so. And I know you probably can't read these, but I'd like to mention Carrie Sang and Catherine Andrews, who have done most of the screening work for animal trials. We'd like to thank Kate Lowe, Fiona Mattia, and Brenda Dobson-Hill. And from Massey University, Associate Professor Andrew Sutherland Smith for help with structural biology. Final slide, I would just like to leave you with some images. On the left, we have some methanogen enzyme crystals before they went off to the Australian synchrotron. In the middle, we have an inhibitor docked into the active site of a methanogen enzyme. And on the right, we have a newly solved enzyme structure involved in methane formation that was first identified 36 years ago, but now the structure can be revealed. And I would like to thank you for your attention.